Good morning, everyone. Thank you for joining us today. My name is Clitel Tailing, and today we will be talking about metagenomic and bioinformatic tools on BaseSpace in particular. And we have Purani Subramanian as a special guest from CosmosID. So let's start. The agenda today will be a brief introduction to shotgun metagenomics, how and why the studies are important, how sequencing is the only solution, and then CosmosID as a software tool solution and how it works. So metagenomics and shotgun metagenomics in particular. It is the study of genetic material directly recovered from any environmental sample. A vast majority of microbial biodiversity has been missed by cultivation-based methods. Recently, the study using shotgun is able to get an unbiased sampling of all the genes from all the members in a sampled community. So what is so important about metagenomic and why do researchers want to do it? Metagenomic studies are usually comparative analysis between metagenomes, which provides an insight into the function of complex microbial communities and their role in the host health, for example. So next generation sequencing is really what started metagenomic studies. 10,000 microbial genomes can be sequenced in a single experiment. Researchers really can't tell which gene belongs to which microbe, but by analyzing the gene collectively, they can learn what the microbes are doing in this environment and have an idea on the status of the environment, healthy versus diseased. To do shotgun metagenomic, really, the sequencing workflow is the following. You can actually extract any DNA from any samples using metagenome DNA kit that we have that is sold by Epicenter. Then the preparation is Nextera XT. You can multiplex really easily using indexing Nextera XT. And then sequencing can be done on the NextSeq, on the HiSeq, depending on the coverage that you're looking for. And then at that point, the data can be analyzed in BaseSpace. And in BaseSpace, you have multiple tools, software tools that are available to you. Now, there is lots of software available out there. And publicly available, you have Megan, for example, which is the metagenome analyzer. It identifies microbial organisms contaminating in sequence study of the DNA and can rapidly compare multiple data sets and classify the functions of genes in metagenome. Kraken is another way of doing metagenomic. It takes short DNA sequencing from metagenomic samples and assigns them to taxa faster than conventionally used program. MGRAST is a web server that allows researchers to obtain comparative and functional analysis of metagenomes. Metavir is another way of doing things, and people can annotate and analyze viral metagenome. HiSeq is a cross-link interaction spot on chromosomes in a genome-1 fashion. So you can study the three-dimensional structure and regulation of genomes. SERPI is another software available, which levels two state-of-the-art aligners for accelerated analysis, SNAP and RapSearch, and which are as accurate as existing bioinformatic tools, but order of magnitude faster in performance. SERPI detects viruses and bacteria by scanning the data sets that can be all the way to 5 million reads. And 5 million reads takes about 5 hours, but 7 million reads takes about 11 minutes. So what we have available on BaseSpace is quite limited. It's not all those softwares that I talked about. But you have Kraken Metagenomics available up there. You have Metaflan. And we also have GENIUS Metagenomics know-how. And now we'll have Purani Subramanian from CosmosID give us a tour of their tool. Juan?
Thanks, Clotilde. And so my company, CosmosID, is a bioinformatics company. We're focused on doing automated and rapid identification of microbes. So today I'll give you a little background on how our software works, and then I'll do a demo of our app in BaseSpace, and then I'll summarize the workflow end to end. So our software is fast, accurate, and easy to use. We've designed our algorithm to analyze the metagenomic sample in minutes. And we've also curated our reference database to remove contaminated sequences and other erroneous metadata so that it's optimized to work well with our algorithm. This ensures that we can do subspecies and sometimes strain level identification, and we can also provide an estimate of the abundance of the taxa in the sample. We've benchmarked our software against a variety of samples, and at the strain level we can get to about 95.5% sensitivity and 99.9% specificity. We also, unlike other conventional microbial methods, if you use shotgun metagenomics and our software together, you can do polymicrobial identification in a single test. You don't need to know what you're looking for before you look for it. You don't need any assumptions before you run our software. So our software can be used to provide results for a lot of different areas, the most obvious of which is infectious disease research. You can do rapid screening for pathogens and commensals, and can also be used in public health for outbreak investigation, including investigation of healthcare-associated infections in clinics and hospitals, and in looking for transmission sources. It can also be used, as Clotilde said, for doing microbiome studies for pharmaceuticals, and also to do environmental monitoring with food safety and water quality. So I'll tell you a little bit about how our database is constructed. So we look at the strains in our database, and we looked for shared regions between the strains here in black, and we look for regions of the genomes that are unique to particular strains here in different colors. And from these unique and shared regions, we collect some marker sequences, and we use those markers to build our database. So when you're in BaseSpace and you have a FASTQ file, we match that file against the markers that are in our database and give you a subspecies or a strain level identification of all of the bacteria that are in your sample. So now I'll go through the workflow in BaseSpace for how to use our app. So when you log into BaseSpace, this is the dashboard page, and you can click on Apps, and that takes you to the page of all of the apps. And from there, you can click on the Metagenomics category on the right, and that will take you to all the metagenomics apps, and ours is right there on the top, GENIUS Metagenomics, and you can click on that. And so that takes you to the page that's an overview of our app. You can read a little bit more about how it works and also get information about how to get help if you need it. But up here on the right is the launch button that you use to launch our app. So from this page, you select which sample that you would like to run our software on. So here I'm going to choose this constructed freshwater sample, and then click Confirm. Then you need to choose which project you'd like to save your results to. So here I'm going to choose this project, Metatest, and then click Confirm. So now we can click on this arrow on the right, and that will start the job. It can take anywhere from 15 to 20 minutes, depending on how many people are also using BaseSpace at the same time as you. And so once it starts running, it will give you a message that it's running. And then you'll get, when it's complete, you'll get an email saying that the job has finished. And if you click on the link in your email, it will take you to a page where it says that now the job is complete. So on the left, if you click on the name of your sample under Analysis Reports, that will take you to the page of results. So at the top of the page of results is our tabular output. It gives you, in the first column, is the list of organisms that we've identified to be in your sample. The second column is the taxonomy of that organism. The third column is the frequency, which is the number of markers from your sample that uniquely match to this particular organism. And the last column is the relative abundance, which is our estimate of how abundant this particular organism is relative to the other organisms that we found. So if you were to sum this column up for your particular sample, it would sum to 100. If you scroll down the results, we have what's called the corona chart, which helps you visualize the composition of your sample. So this particular sample is a laboratory constructed sample, and it's modeled after a real sample from the Delaware River. So if you click on, in this case, a phylum like proteobacteria, you can expand that out and examine just this particular phyla. In this case, it contains a class of gamma proteobacteria and some Enterobacteriaceae, which are known to be pathogenic. If you scroll down again further, we have a tree view, so you can view the taxonomy of your organisms in a tree if you want to see how related or unrelated the organisms in your sample are. You can expand the node. So, for example, if we look at this genus of Escherichia, you can, click on the node to expand and see all of the organisms under that particular genus that are in your sample. If you click on Output Files on the left and then on the folder for your sample, you can get a list of files that you can download of the output of our app. So there's the table output, which is a tab-separated file, and then the tree and the corona chart can download as HTML files. And you can save those as image files from your browser and use them in presentations or publications. So right now, our app in BaseSpace is free for you to use, and we encourage everyone to go and try it out. If you need some data to test it out with, you can go to Illumina's or BaseSpace's public data repository up top here and click on Metagenomics. And this bottom set, this third set of samples, contains that constructed freshwater sample that I just showed you, and it also contains a sample from the Human Microbiome Project, which you can import to a project of your own and test out our app with. So now I'll give you a summary of the workflow, which is to do the DNA prep and use the Illumina sequencer, and then to upload your data into BaseSpace, and then you can use our app to get the results of the analysis of your metagenomic sample. And it's as you saw, it's pretty easy to use and very straightforward way to get your results. So if you have any questions, even after this seminar, you can email us or visit us at help@cosmosid.com or visit our website at www.cosmosid.com. And thanks to Illumina, and especially Clotilde and Raymond and Mike for giving us this opportunity to show you how to use our app.
Purani. Thank you, Purani. We have quite a few questions. Actually, I have one on my own, which is, what database does GENIUS use?
So our current app in BaseSpace is only for bacteria. It has about 4,700 bacterial strains. And internally, we're working on expanding that to be, I think our next database will have around 31,000 strains. But currently in BaseSpace, we only have bacterial database. In-house, we have other databases that, such as fungal and parasite and DNA viruses, but that's not yet deployed to BaseSpace.
Okay. Another question that is coming from our public is, could CosmosID be used for 16S metagenomic data? And what could be the major limitation if doing that?
So no, it's not designed to work with 16S data. It's only designed to work with WGS data. We can do much more specific results than 16S using WGS data.
Okay, and then could the software break down to serotypes?
Yes, we can. It depends on how clonal the organisms are, but yes, we have those individually in our database.
Good. And then another one is, what is the most key point for a high-quality meta-sequencing? And then the question is DNA quality. Definitely, DNA quality is important to put into the sequencer. That's definitely one thing. And so by following the workflow that I actually said, it definitely helped. But then afterwards, do you think, Purani, do you think that coverage makes a huge difference in your software also?
Yes, we usually recommend around 10 million reads minimum to get a good result. And you also have to be careful about the amount of host background that you have. If it's, say, a human clinical sample, and if there's a lot of the host background DNA is very dominant in the sample, like 95% or something, then you may want to do some host DNA reduction so that you get better resolution of the microbial content that's in the sample. It also depends on, one more thing I didn't say, it also depends on how complex your sample is and on the diversity of the sample itself.
And actually talking about diversity, do you have, is your database comprising maybe the microbial that you find in human microbiome versus environmental, and do you have both in the database?
We have both. Our current database does have most of the major organisms from the human microbiome, and we have some environmental organisms, but we're also expanding our database to include a wider range of more environmental organisms as well.
Nice. There's another question that says, can you search the data to quantitate bacteria with a particular target sequence?
So you already know beforehand what particular sequence you're looking for, and not like a whole, and not a particular strain. So currently, as the app is implemented in BaseSpace, no, we choose the markers for you in order to do the best job of identifying the bacteria. You can't look for a certain subsequence that you particularly want.
Okay, another interesting question is there any way to compare one particular bacteria between groups by using your software?
If you want to, between two different samples?
Maybe, yeah, we could put it this way. Sounds good.
I mean, you can generate the results, and you can, if you download the results, you can do the comparison between the two samples to see if they contain the same organism. And our implementation in BaseSpace, we don't currently have data visualization tool to show you to do direct comparison between the two sets of results, other than just downloading it and looking at it yourself. In-house, we have some tools for that, for our in-house version, but not for the version in BaseSpace yet. But thanks for we'd also like any feedback that people have for using our app for features that they would like.
I'm looking through the questions, trying to find you another one. Is the software good for soil metagenomic?
It's okay. We, like I said before, our current database mostly is skewed towards clinical application like human microbiome, although now we're expanding further. So we have done some analysis in the past, and so it depends on the diversity of the sample. But you can definitely get at least some idea of what's in the sample. We may not be able to go down to as specific as we would be able to with other kinds of samples.
Okay.
But we're working on that, on expanding our database for environmental. So.
Good. How many reads was in that example sample that you showed us, and how long did it take to analyze this example?
I think that sample had about 30 million reads, and so in the actual runtime, so when you submit a job in BaseSpace, there's a pending execution until it starts running, and so from the time that it starts running till the time it finished was I think less than 10 minutes, maybe around 9 minutes it took to run.
Okay, thank you. It says, where in the app do you list the filters employed for quality control of the FASTQ files before they are compared to the database?
So we do all the filtering and analysis on the back end, and we currently don't show that those parameters to the user in BaseSpace. We just we have the simplest version of our app and our algorithm deployed there. Maybe in the future we may open that up for the user to use, but right now we do the filtering for you.
Okay, another nice question is, when you say bacteria domain, do you include also archaea in there?
Yes, we do.
Okay. And I think that whatever questions I skipped, we will definitely get back to you through email. Purani will have all those questions, and we'll be able to get back to you. I do want to thank you, Purani, for sampling this wonderful app, and I can't wait to try it myself. Thank you for everyone to joining us, and I want to say that at the end we will share our recording, and we will send an email with the link of the recording in there. So thank you for joining us this morning. Purani, do you have anything else to add?
No, thank you so much for this opportunity, and if anyone has any more questions, don't feel free to contact us as well. Thank you.
Thank you.